So before we begin, I'd like to take a moment for us all to acknowledge the traditional owners and their custodianship of the lands on which we're meeting today, the Turrbal and Yagara peoples. And I would like to acknowledge and pay my respects to their ancestors and their descendants who continue cultural and spiritual connections to country. I think when we're talking about science and environmental stewardship, it's always really important to remember and think about how much we have to learn from respecting different ways of knowing, including from the first scientists of this country. So I would like to announce Dr. Hannah Fraser, who joins us today from Melbourne. Hannah Fraser did her PhD at the University of Melbourne as part of the Centre of Excellence for Environmental Decisions, where she worked to understand and classify and protect Australia's woodland birds. Since then, she has been working in meta-research and open science, trying to understand how to make the findings of ecological studies more reliable and robust. She is now returning to ecology and is excited to be studying birds again with the Marin Ecology and Conservation Policy Lab and is based in Perth. Thank you, Hannah.
I'm hopefully visible now. Thank you so much for having me today. I'm going to tell you a bit about some of the meta research that I've been doing. For this same data, different analysts, variation in effect size due to analytic decisions in ecology and evolution project, which has been one of those projects that hangs around for 1,000 years and hopefully will finally be published soon. Here we go. So when you look at studies on similar topics, you find a lot of heterogeneity between studies. And this is a meta-analysis of meta-analyses, as obviously look at how much variation you get on average between different studies within a meta-analysis. And it's this I-squared value, which is the proportion of variance attributed to difference among studies, is 85 to 90%. And this is, there's a few different types of heterogeneity that play into this. There's biological heterogeneity, methodological, and analytic. Biological heterogeneity is the one that we think about, basically, when we're doing a meta-analysis. We usually attribute almost everything to this. So it's the idea that the environment and the different contexts and the different species between different studies is driving the main part of the difference between studies. And that's what we hope as ecologists, because we hope we're finding mostly things that are true about the environment when we're doing a study. On top of this, there's also differences that you can attribute to differences in your study design, so how you choose your sites and what methods you employ. That's going to make some difference to the kinds of results you get. Large-scale collaborative projects like NutNet aim to try and combat this by standardizing approaches across a number of different research groups study the same question. And when data is collected in the same way, it sort of at least minimizes the effect of methodological heterogeneity. And then there's this analytic heterogeneity part, which I think we don't really think about, or at least I never used to. So the idea of this garden of forking paths where people can make a number of different decisions along the way and end up theoretically in quite a different place. And the number of different analytic choices you can make is a function of how specific your research question is, how many variables you collected, and whether you pre-registered or made a plan, a specific plan for what you're going to do before you started looking at the data. So many analyst projects have been a thing in a few other disciplines, and we've sort of tried to roll out that approach in ecology and evolution to see if the similar things hold true. So the basic idea is we get a whole lot of researchers to look at the same data and answer the same research question, and we see whether and to what extent their answers differ. The first of these projects was done in social science. Broadly, the question they looked at is, are soccer players with dark skin tone more likely than those with light skin tone to receive red cards from a race? And you can see they all analyse the same data set, and there's quite a lot of variation in the results they got. There's a similar study in looking at, does immigration reduce support for social policies among the public? And as you can see again, there's quite a lot of variation in what people are finding. So over here, there's sort of a negative response, and over here there's a positive response, and there are these, the grey ones are a non-significant response. So you're getting a really big variation in the overall message of those papers, what that paper would say, depending on who did the analysis and how they did it. The same again here, looking at estimating the effect of compulsory education on the proportion of teen pregnancies, and again, we've got responses that range from significantly negative, significantly positive, and everything uncertainty in the middle. So we wanted to look at how much this is also true in ecology. And we wanted to look at how much do our analyses affect the results we get? And are these differences driven by the quality of the analysis, the uniqueness of the variables we chose, or the choice to include random effects in the structure, which sort of is an index of quality, depending on the data sets you're looking at. And then because it's sort of a, possibly some research, some data sets are a all roads lead to Rome situation, we had to look at 2 different data sets to make sure that there's some amount of generalizability, though obviously it's sample size of 2. So should we expect there to be lots of analytic heterogeneity? And I would say yes. We've got broadly stated hypotheses, we've got opportunistically collecting lots of variables, we've got complex modelling approaches that require lots of different decisions to be made. So I would expect that we're going to have a similar problem, and that's what we expected going into this. We looked at 2 different questions. So the first one is, to what extent is the growth of nestling blue tits influenced by competition with siblings? And we got them lots of different variables. We've got 452 broods of 332, of which were part of a brood manipulation, and we've got 2 different outcome variables and a whole lot of different predictor variables and covariates that people could choose. And we recruited 78 teams who submitted a total of 174 distinct analyses of that question. Lots of people analyse things in a couple of different ways, which is, I think, pretty typical, but also not something we had planned for, and we had to do a whole lot of shenanigans. We were just like, well, everybody's just going to submit one analysis, obviously. We forgot about how science works. Backwards rather than forwards. Here we go. And this is the different outcomes that people gave to that question. So we've got, on the left here, we've got a relationship where more siblings are associated with faster growth, and on the right, more siblings, slower growth. And we've got a small number, admittedly, over on this less plausible negative positive relationship side, and the majority here on a where you might be expected to be side, but still a massive variation in the magnitude of effects, even if you just look at the ones in the plausible realm. We also asked people to give us a verbal statement about what they think the overall response is from their analyses. And based on those, we've got 5 who thought there was a mixed effect. So like sometimes you'd see a negative effect and sometimes you see a positive, depending on your analysis. 37 plus 27, probably should have done that math beforehand, because there was a negative relationship. And some of those were qualified by certain clauses and other ones were written in a really certain way. 5 of them found no significant effect, and no one said overall there was a positive effect. So these people had presumably also done other analyses over this side and in the mixed or qualified categories. The other question we had was, how does grass cover influence eucalyptus species seedling recruitment? And we had a similar kind of data set, quite a few different sites, and ambiguous outcome variable where you could come up with your own based on different seedling size categories and predictor variables, including 4 different grass cover categories and all these different covariates related to basically all the things we would normally kind of have coordinates, seed and ground cover categories. And we had 68 teams of analysts who submitted 128 distinct analyses of this as well. And you can see that we got sort of less variation and sort of more. Yes. So we've got this lovely outlier who was speaking a completely different language to everybody else. And then over here, you can see that everything's a bit tighter on 0. And that's because the overall effect here is probably quite close to 0. So, but because it's quite close to 0, we've actually got more difference in the number of people who, like, we've got more of a spread between positive and negative, but they're only like within a reasonably narrow band. So we've got 6 teams said that there was an overall positive effect. Most of the teams said basically not really much of an effect, and 18 teams concluded there was a negative effect, and then there was 8 who did a mixed effect, so that there was a mixed effect depending on which variables you included. So basically, we don't know is the answer to this. Depending on who you ask and whose analysis you did, you could get any answer for this question. So, is the difference in these results driven by analysis quality? We had each of the analyses that people submitted reviewed by other researchers part of the project, and they got a score on the scale of 0 to 100. And it didn't really predict difference from the mean estimate, which is what we're looking at here. So we looked at how different each of the analyses weren't from the mean of everybody's as an index of like how wildly different they might be, and it didn't correlate very well at all with rating of how well the analysis was done. Was it driven by the uniqueness of variables selected? No. So we used Sorensen diversity index, where we, instead of looking at species, we used variables selected to calculate a mean uniqueness of the sets of variables used for each of the analyses. And we had a look at how that correlates with, again, the deviation from the meta-analytic mean, and there wasn't a lot going on there either. So not convincingly driven by uniqueness of the variables we used. Inclusion of random effects. So only 3 blue tit analyses didn't include random effects, so we didn't analyze that, as you might suggest. And for Euclid's, not convincingly. It's one of those really unsatisfying studies where there's no no specific thing. So is it driven by outliers? In the case of Euclid's, as you might suggest, the overall picture is substantially impacted if you get rid of this fellow and his friends. But the overall pattern of there being a significant amount of variation in the results remains. The blue tit analysis, there weren't really substantial outliers, so removing them did very little in terms of explaining the pattern. So what might be driving the differences if it's not these things? So it could be a cumulative effect of a whole bunch of different decisions, none of which are necessarily indicators of good or poor quality. It could be analysing subsets of the data. So for the Euclid one, that extreme outlier is an instance where people had massively subset the data to only include certain parts, which can obviously lead to quite different results to everybody else. It could be the inclusion or exclusion of some specific variables that had a lot of influence. Though we had a casual look at this and it didn't seem to be obviously the case. Perhaps if we dig further, we'll find some evidence of that. And there's a thought that data sets with a stronger effect, like the blue tit data, may see more variation than ones with a smaller effect, just because there's more sort of room for there to be a trend detected. Whereas in the Euclid one, there's not a lot going on. So, why should we be worried? Well, probably being worried isn't especially helpful, but you know what I mean. So the results of our analyses, or the analyses we see in the literature, might be driven more by analytic choices than by the ecological situation that we're trying to understand. And the degree of variation possible between these results leaves a substantial opportunity for questionable research practices to influence the overall result, which can lead to biases in the literature. Just a little snippet on questionable research practices that some people might have seen before. We did a study asking people about which practices they'd engaged in the ecological community that might influence the results of their papers to be more exciting and more certain than is warranted by their methods and their outcomes. So these are some of the things we looked at. Today I'll just mention the cherry picking ones, which is essentially when you conduct a whole bunch of analyses and then only present the good ones. You can see that similar to other fields, these ones boxed in orange are different versions of doing lots of analyses and only presenting the good ones. And about 50 to 60% of ecologists say that they do this, which is pretty similar to education and substantially more than some of the other samples, which doesn't surprise me again because we're collecting so many variables reasonably opportunistically because it's so expensive to collect data that we just collect everything we can for our often. But yeah, so this means that if you conduct a whole lot of different studies, eventually you'll find something good if you conduct enough analyses. So this is the whole idea of doing p-value stuff 5% of the time you'll find something. Doesn't matter if it's there or not. And this is a lovely XKCD thing I use all the time because it's so good. Yeah, so it increases, this kind of practice increases the number of false positives in the literature. So basically that's where we say that we found a true effect and actually it's sampling bias or random variability that we're presenting as if it's a real ecological thing. And it makes our research look more certain than warranted. And what can we do about it? So there are a whole lot of different things, none of which is a complete solution and none of which is necessarily applicable to all circumstances. But one of the main things for addressing questionable research practices is specifying our research questions precisely. So if we link our research question to the outcome measures and the variables we're using, that provides less scope to vary that to answer different questions, or at least it makes it more transparent exactly what we're looking at. Pre-specified analysis plans, that's where you publicly or at least privately write down in detail what you're going to do before you do it. So I think the thing with this is that people aren't doing it intending to end up somewhere different than when they started. But it's easy to lose track of exactly what you planned and get excited about this new method that is providing better results. And if I started with it, it would have been the same. So it can keep you on track with yourself and it also makes things much more transparent to pre-specify things beforehand. It's pre-registration in general is a practice where you can do this online and it gets a timestamp and it's unalterable and people can refer back to it. But even I think just writing it down and sharing it with your supervisor, super useful. Robustness checks and sensitivity analysis to look at a number of different ways of doing analysis and seeing which parts affect your outcome and then describing that ideally in your manuscript is a really important step. And I think a lot of people do sensitivity analysis all the time, but it's not necessarily the thing that makes it into your final manuscript because it ends up making less of a neat story. Model averaging, if you're conducting a number of different analyses, model averaging is an option for combining them together and getting a less extreme, more modeling effect. I don't like this as an option. Multiverse analysis is basically a similar thing to the many analysts project that we just did, except you do it all yourself. Basically, you come up with as many different ways of analysing the data as you can and see how much it varies. Obviously, there's a significant amount of work in that. And there's a specification curve approach, which is basically the same as a meta and multiverse approach, that you keep track of exactly which things affected it, and then you plot it out so that you can see the patterns in it more easily. Right, this is my favourite and least satisfying recommendation. Perhaps we should just be more cautious in how we interpret and describe our research. It's not really how things are set up. People don't like reading research that's presented in a cautious and understated way, and it's not judged well. And it's really easy to non-critically read the literature. But I think that this would get us a lot closer to somewhere sensible. Just being a little bit more circumspect when we're describing things and a little bit more skeptical when we're reading them. All right, so analytic choices are really important in driving the results that we're seeing in the literature and the results we're creating ourselves, and we need to be careful about that. But I think recognising that that's a factor and a problem is an important first step in moderating the effect of that on our understanding of ecology.
Yeah. Hi, that was fabulous. Thank you. And it's great to see this work done in this discipline. Did you look at social characteristics of your analytical team and whether that contributed like duration, like length of experience or disciplinary, like even in social science, there's this enormous disciplinary variation in analytical approaches. And did you look at the social stuff?
So we don't have disciplinary, but we do have experience levels. And it's tricky, obviously, because the teams have different numbers of people, and just having one really experienced person and a bunch of inexperienced people mean that you're an experienced team. That's sort of how we've taken it. It didn't make a substantial difference when we just looked at the experienced teams. But the disciplinary thing is something, the domain expertise thing is something that we're really interested in. And my colleagues have just got a grant to continue this work and look in more detail at that over the next 3 years or something under the NSF.
That would be fabulous. And things like tenured versus non-tenured and things like that, whether there's really subtle effects on how we focus and are comfortable with non-significant findings.
Yeah, definitely.
Great talk, Hannah. Thank you so much. I have multiple questions. I'll try to keep it simple. One is a follow-on because I'm curious at the size of the team.
Yeah.
So it's like one person, obviously much easier to be like, I think this and just move it forward. Is that no?
We haven't looked at the size of the team. I think probably half of the teams-ish with just one person.
Yeah.
And then the others were quite variable. Usually like a senior person and their students doing it as an extension activity, as a common format. But definitely there will have been dynamics there.
Okay, so the thing I'm more interested in was where you evaluated the studies and gave them a ranking from 0 to 100 in terms of whether they were crap or not.
Yeah.
So what are the characteristics of the crap studies? Like how did they determine that they were so bad? Who did them?
It's all a little bit so we followed just sort of a standard peer review approach where people used their discretion to determine what constituted a good or a poor analysis. There was extremely low agreement among reviewers about what constituted good versus poor analysis. So I can't really say much, but possibly that's what you'd expect. Definitely increased my doubt in the peer review process. Yeah, people can't agree at all on what constitutes a bad analysis. There were some people doing reasonably out there things, and we did a version of this analysis that excluded those. And didn't make a whole lot of difference.
So this is actually the result I'm really curious about, because does that mean that we can just like run 100 crap analyses that we don't spend a lot of time worrying about whether they're specified correctly? And that's gonna give us the range that will be able to tell us the degree to which the effect size might fall within, rather than being really worried about getting it exactly right? Like if crap studies didn't affect this at all, and it's just actually the amount of studies that give you the range, then I feel like we're spending too much time.
I mean, that feels like a dangerous, but and not no,
Yeah.
right?
It's the number of studies, not necessarily the quality of the study, that are gonna give you the range of that effect size.
Definitely in terms of scoping out plausible space, I think overthinking exactly which variables you include takes you down a particular path where actually a number of different options were equally plausible. And yeah, so throwing a bunch of things at the wall and then also maybe having your ideal, I think this analysis is right, analysis is probably a good approach. Like, I think this is the answer, and now I'll do a whole lot of other things and see where the like what range of things is plausible.
I see a new CBCS tagline here. More crap studies.
Yeah.
Great talk, really reminds me of the stuff I read weekly on Retraction Watch.
Oh, yeah.
Which everyone should read, you don't know about it.
It's so depressing though.
I'm actually not in ecology, I'm a molecular biologist, but data is data. So, and the nature of all of these models is that all these statistical models have assumptions which are not necessarily perfectly fitting of any type of real life data. So we generally try to make things that fit these models. And to do that, we often use transformations to do that. What role did transformations play in any of this? Surely that changes the effect size massively, maybe not the direction, but the number, and especially if you consider things which are logic or not logic.
Yeah.
Sorry.
Yeah, so we haven't looked at it explicitly. We had a subset of analyses used transformations. And then on top of that, there's also substantial difference in the number of the link functions that people use, which is essentially the same thing. Yeah, it definitely be worth unpicking. The transformation thing is like a classic ecology, just log it and it'll situation. I don't know if everybody else has heard that. I don't know. Putting my foot in it. Anyway, that's an approach I've heard, not from people I work with. But, yeah, I think transformations play a huge role. It's just an extra thing that you can try to make it work, which multiplies the effect of different analytics.
It also makes things less comfortable between them.
Yeah, exactly.
Hey Hannah, great talk. I got two questions generally, hopefully they're short. The first one is, how can you tell if a study is doing cherry picking? Obviously, they don't say we cherry pick the data. That's probably the first. And the second one is, do you know whether the quality of the journal is in line with the quality of the analysis? Did any of that come out in your work?
Not in this stuff. Typically, the more prestigious the journal, the more likely it is that you did cherry picking or other questionable research practices, because it really tidies up your analysis and makes it look much more powerful. Ones where you're presenting a nuanced view, where you conducted a number of different analyses and some of them showed a strong effect and some of them didn't show a strong effect, those are more likely to end up in a less prestigious journal, unfortunately. That's getting less true. They've reduced the word count, like they've gotten rid of the word count for the methods in a number of different journals, prestigious journals, which makes a difference in describing having included sensitivity analysis and stuff. I think, yeah, you can't really tell if someone's done cherry picking. If someone's done a study that doesn't include any kind of sensitivity analysis or multiple analyses, perhaps they only did one analysis, but that would be a reasonably common thing in ecology in my experience. I think people usually look at things in a number of different ways to get an understanding of what's going on. And if that's not presented in the study, I would suspect that there was stuff that hadn't been written, but you can't.
Thanks, Hannah, that was great. So this sort of movement of pre-registering and looking at questionable research practices sort of started in psychology, is that right?
Yeah.
So given that they're how many years kind of ahead of us in this reckoning with the field? Like, are there things that we can learn from them? Like, how much of a crisis did this cause in psychology, and how can we, I mean, obviously you had your lessons at the end, so those are part of that, but are there any things as like an entire discipline that we should be, I guess, looking out for or watching that came out of earlier movements?
I think the biggest thing is just recognizing that this stuff is going on. I think that it's something that, in my experience, ecologists don't think about a lot because we're really focused on understanding the environment and we don't care about what's going on in psychology and what the implications are there. And there's a tendency to think that psychological research is maybe a little bit woollier than ecological research and disregard things there. So I think the largest thing from my perspective is an awareness raising component, because most of it's just actually just be a bit more careful, just document your stuff, say what you did. And they're things that we all know that we should be doing, but it's counter to the incentives of the field where you also need to present a really clear, clean story to get published, to get grants, to get, so enough awareness raising to change the way people write stuff so that the norm is more nuanced than it is now, I think.
Sorry. Yeah. Sorry, Saxoras, this is super interesting. So your recommendation is essentially about changing future practices. But can you make any suggestions for evidence synthesis? You know, given there's this variation, can you do anything to account for this kind of issue when, for example, doing meta-analysis or?
It's tricky because it's changed the studies that occur in the literature. In other fields, like in medicine and things, where people have to pre-register everything that's in a clinical trial, you can look at trial registrations and find out what their results are and compare that with the overall thing. That's not possible in ecology because pre-registering is not a thing and people don't publish research that's not going to get them anywhere because we're busy and not underfunded. You can do things like P-curve analysis, which gives you an idea of how biased the results are likely to be. So that looks at the distribution of p-values you get, and if you're getting a whole lot that are just under the 0.05, that's an indication that people have been doing some things to make their results fall into a significant category. It's trickier in some fields of ecology when we're not really looking at p-values. So I don't have a good answer, unfortunately.
It's such a wonderful talk, and there's so many things that I could potentially ask. It's really amazing talk, actually. But I was wondering, have you thought at all about, because like there's that, you said there's the subjective score of quality, and that after looking at all of them, there was so much disagreement between the reviewers as to what was good and what was bad. Have you thought about like trying to backcast a power analysis of all the studies and then plot like the power of each study? I guess some of the studies would be harder to do power analyses than others. It would be massively time consuming.
It'd be really interesting. We looked at the effective sample size, which is not really the same, but it depends on how many variables you've got. I would say that all of them were underpowered as a take. That's pretty typical in ecology. There are lots of variables, not very many samples. But it'd be really interesting to have a look at if we had all the time.
First of all, I'm really amazed that you got that many people to come and do an analysis for you. How did you recruit people? Like, what did you ask them, and how did you find the group of people? Who were they?
So it was the middle of a pandemic, so I think everybody was a little bit typical. Yeah, have a pandemic. I think most of the people were recruited just from Twitter, as it was then called. I don't know whether you have any success with X now because people are disillusioned and not looking there as much. And then we also did some mailing lists and things, but people it's a pretty appealing prospect, really. It's like, hey, do you want to do a quick analysis and get a publication that's going to be widely cited?
So they had to be, what were your requirements? Could any old rando say, yeah, I'll do an analysis, or did it have to be
They had to
someone
be-
working in the field, or?
I had to be broadly anybody who considered themselves an ecologist. So they didn't have to be domain experts.
And you just asked them, like, you didn't give them any guidance, it's just like, here's the data, like, you didn't say, I'd actually like to get a whole range of different ways, so feel free to do something crazy, you said, do your best, do what you would do. Is that what you told them?
Analyze this as if it was your research question. Here's a research question, here's the data.
And then I find it amazing that it looked like every single answer was different, like there weren't
So
even
nobody
clumps of
did,
answers.
nobody did exactly the
How
same
could that many
analysis.
different ways of analysing?
It's horrifying, isn't it?
I don't get it.
Yeah. Yeah, there's just so many options. Garden of forking paths.
There you go. And then, how many different ways did you analyse your data?
Well, I mean, many more than we should have. So we pre-registered what we were doing. We did it all as a registered report. And then after we'd done it, we added a whole lot of additional post hoc analyses that I really wish we hadn't done, because they were a nightmare and didn't reveal anything new. Anyway. So we analysed it as we'd said, and then we also did a whole lot of different subsetting afterwards to see, like, what if you exclude these people, and what if you only look at people who are domain experts, and what if you only, none of that made a difference, and it just took a really long time.
And that wasn't in the pre-registered.
No, so we just
Do that,
pre-registered
but-
the ones that we described here, and then everything else is presented as exploratory research.
I guess one of the things with the shift in biomedical stuff and mandatory pre-registering and able to publish, one of the things that I guess that happened in parallel at that time, and I was working in clinical trials at that time, so was that there was a much bigger culture where journals said we are keen to publish your negative or null finding studies, as long as they're of adequate quality. And if a null finding can be interpreted, it needs to be published to try and remove that pressure for the positive finding. I know I'm not an ecologist, so I guess the challenge in ecology, if we're thinking about the push-pull of this push towards a cherry picking or p-hacking, even if it's subconscious at times, is what do we do with null findings? And I think, and as a field, as ecologists and biologists, I know interpreting null findings is much more complicated than it is for social scientists, so that's a little benefit for us. But I think as a field, knowing what to do with that body of work and how to interpret it, is it something you can learn from, or does it just mean your method stuffed up? Or, all of that stuff, I think the field really needs to sort that out in parallel to this other cultural shift. I don't know how to do that, but.
I think that's a really good point. And I think the tough thing from my perspective is prioritizing doing that. So I think most people you talk to would be like, yeah, no, I'd like to publish all of the stuff I do, but we're really time limited. And as soon as you find something that's non-significant, it's hard to interpret, and you're not excited about, it just drops down your priority list, and you do the next important thing that you've got that's already past you, and it's hard. It's just hard. It's hard to do. Needs some kind of cultural shift. I completely agree.
Yeah, and that excitement factor is also, like, I look at the things on my list to publish, and the excitement factor is what drives what gets to the top of the list. And so there's another layer of bias in addition to stuff.
One thing I've seen, I think Mark Bergman's been driving with conservation biology, is the idea of just like publishing a minimum thing about your finding, just like a short snippet, rather than having to go to the effort of writing up a whole paper. And that's maybe something that could contribute.
Yeah, right.
And I was just wondering on that Gelman quote, Garden of Forking Paths. Do you think we might get to a point where we actually, in papers, have to illustrate that path? Because I know in my own stuff, I've done analysis and I've looked at residuals and gone, oh, let's go. And so you backtrack and you try something else. And might that be something that we could do to formalise the approach that we took?
I think it definitely is a possibility. It's sort of like what you do in a metaanalysis, where you talk about all the different steps you made to exclude different data. I've never seen it done for analysis, and it might be a nightmare, but it would be really helpful.
Yeah.
It'd be a lot more readable than like describing
Yeah,
in the methods every
yeah.
single thing you did. And
Yeah, definitely.
I have a personal question that you might not be able to answer now, we might need you to come back in 5 years to answer, which is sort of, again, two parts. One, knowing what you know now and having done this, does it make doing science or publishing less fun for you? And two, this is the question that you would only be able to answer in the future. Do you find collaborations harder because other people want that nice storyline, and you're the one that's like, come on, people, like, we could do this another way, it's gonna tell us this other thing, we could do it this way, it's gonna tell us another thing, we can't actually say this, and they're like, we have to say something. So does that is that making collaboration for you harder at this point?
Definitely the collaboration thing is a barrier, particularly I think it's more problematic in an ecological space because this is so new and a lot of people don't subscribe to the whole idea of being more transparent, and it's counter to the way things have been done for so long. So it feels like an uphill battle, even when I'm working with other people who are meta researchers in ecology. I'm still constantly saying, no, we pre-registered that, it has to go in a manuscript. And that's frustrating. But I think, over time that'll become less true. In terms of doing research on my own or mainly motivated by me, I actually find this more fun. I love writing pre-registration. It makes me feel like I'm doing real science. I made a research plan and then I'm going to find out if it comes true rather than sort of feeling my way through it. I find that really satisfying personally, but maybe I'm a weirdo. It does mean front loading a lot more of the work.
I'm curious, Daniel. I mean, like, you're more experienced than me, but isn't it when you get non-significant findings, you're like, okay, what's this thing? Why is this mysteriously different from everything else? And then you question, and then you put this tag at the end of your discussions, like, this is non-significant, we don't know why, therefore we need more research, therefore give us more money. I mean, that's a highlight thing. Like, we don't know why this is not significant. This is important, and hence a source for more funding.
Yeah, the real answer is that you get to a certain point and you just write perspectives and reviews, so therefore you don't have to answer any of those questions. You're absolutely right.
Yeah, I think that's a really good point, because I think some of the most interesting findings that I've been involved with are things that, because you're not asking the question unless you think something's going on. So if it's not, then that's weird. That's also interesting, and I think that's one of the things we're lucky in ecology to have that. I mean, Courtney, your research is a really good example of that, one of the examples I'm thinking of, and some of the most interesting papers have been null results.
Does that not support funding? Like, saying that, oh, we only have limited funding to say that this part, we don't have extra funding for this weird part. And
But
does
that's the
that,
mystery. What's the mystery? We haven't solved this mystery yet.
yeah, exactly, does that support, and give us more money and help my career?
If you're good at writing grants, it's obvious. If you're not good at writing grants, it looks like you haven't been successful. It just all depends on the spin.
And it's that interpretability of a null finding, like if it's a bloody well done study.
Yeah.
And you've gone, this isn't doing the thing, like that's great, but if it's like, oh, is it because I just mentioned that in this way rather
My
than
sample
that
size
way?
wasn't high enough.
Is it this, as soon as there's doubt as to why there's a null finding, like from a methodological perspective, it's really tricky, but.
Yeah.
Great talk. So, so in human genetics and genes of data sets and those two diseases. So after we get a data set, the genetic data set for the human disease, normally we have a listing pipeline to create. So depending on the research questions. So it'd be better to have existing pipeline ecology than be easier for us to go through. So for each step, you can have different choices. You can select different tools to data pre-processing or things like that. That being considered because we don't need to make so much decisions. So yeah,
Yeah, I think that's one of the really great things in the molecular space is that they're actually doing a whole lot of practices that already limit the kinds of decisions you make. I think often molecular and other lab-based work does a lot more record keeping as well. Is that right? Do you have lab books that you have to keep as well?
Why that people so regularly to data analysis, so I'm not sure if they're sure about the lab issues.
Yeah, cool.
Yeah.
But the people in wet labs also do that, and that's another thing that's really great for transparency and openness. I think, yes, having protocols for doing analysis can be really helpful. I think it's a hard thing to convince people to do though, because there's always I think the classic ecological response to anything is it depends. And I think that that's prevalent through anything. I think that there'd be a lot of resistance to constraining analytic choices because it depends. Like in my circumstance, I need a different thing. And I think that there'd be a lot of value in having protocols. But I'm not sure how we could convince people to adopt them because it's quite culture and contrary to the culture in ecology as I understand it. That's just my
And
That's
it's
space.
also dependent on your animal if you're a field ecologist, whether
Yeah.
they show up or not, whether and when, and it's just nature's so unpredictable.
Yeah, I mean, in theory, you could, like, one of the ideas in pre-registration is that you plan all of that stuff beforehand to as much as you can. But our ability to do that is limited, and our desire to do that is also limited, which is tricky.
Are there any ecology journals that are incentivizing pre-registration in a way that's good for authors in the sense that, like, for example, you could just incentivize registration generally, but that, like, then no one's probably gonna do it. But one way you might incentivize is say, we will peer review your pre-registration, and if we like it, we guarantee we'll publish the results, even if it's null or not. And I think there's maybe other fields that you do something like that.
Yeah, definitely. So that's called a registered report, and there's a few, like there's most, or maybe not most, a lot of psychology journals do it. And I think there is maybe 20 ecology journals that do it. The uptake has been rubbish. But it's the kind of thing that there has to be a long lead time on, obviously, because you have to pre-register before you've done the research, and the research always takes a really long time. So I think it's going to take a long time. Conservation biology does registered reports. And there's a whole list of, but yeah, some of the major journals are doing it. I think Nature, Ecology and Evolution are introducing them as well, or already have. So there's a few really good journals that do it, but it's a big commitment upfront for authors and yeah.
But how much of a commitment from the journal is for them to weasel their way out of publishing something if they don't like the answer?
I think that they're pretty committed to it. I mean, it's not much of a commitment at the moment, obviously, because they'll get like 2.
Right.
And they're like, that's just not gonna really hurt them particularly, but in theory, they're committed. I haven't heard of a circumstance where people have reneged. No. Essentially, if you do what you say you did, say you're gonna do, they have to publish.
So this is a very library question following on from that, but if you're doing that pre-registration process, is that the point at which the journal starts asking you for article processing charges?
That's a very good question. I don't think that it corresponds to that. Though I can definitely see that being down the track.
So
No, I've got a lot of questions. It's the best part.
I'm curious about, so one of the big issues in ecology is the cost of collecting the data, right? So we're constantly reusing data. I personally have. I was gonna say never, but that's not true. I did collect data on the size of scallops and the growth of scallops at one point in time in my life. But other than that, I've never, ever collected data, right? So how does the reuse of data align with the need to pre-tell people what you're gonna pre-register, what you're going to do, when you're just like using other data sets, they collected it for a totally different reason.
Yeah, it's tricky because it's very hard to be completely blind to what the data set says before you start your analysis. The idea of pre-registration of existing data would be that you haven't looked at the data before, like you haven't touched it, you haven't touched the data before you pre-register what you're going to do. So that if you've read an article based on that data, you're already influenced a bit. But I think that space is, tricky, but possible to still constrain things and get some transparency. Just maybe not completely. I feel like there was another hand.
We need to check online. Okay. That's just compliments online. No, I did it, no one else.
Yeah.
I'm just wondering if the advantage, so I love hearing that you enjoy the pre-registration process, because that's very heartening, because it sounds like just a pain in the ass. But I wonder if having this like upfront workload, have you found that makes things easier down the track? Like I often feel like by the time you get to the end of analyses and you're writing the paper, you kind of just wish you were dead. The sort of getting used to that, even worse.
Yeah, definitely. We've all been there.
Yeah. And so do you find that having to really think through that plan and do all that work at the beginning sort of evens out the workload throughout the process, or even makes it easier towards the end when you're losing steam?
It definitely makes it easier towards the end, particularly if you do a registered report,
Yeah.
because in that instance, you've already written your introduction and your methods and they're peer reviewed and you can't change them. Like you can add extra things to your methods, but those parts are locked in and you don't have to worry about them anymore. Which is lovely. Even if you do like a standard pre-registration where you just write out your methods in detail, you don't have to write your methods again. You just,
The worst part.
you already did that part. Obviously the rest of it's, but because you put more thought into exactly how you're going to answer the questions, I think,
And your question.
and your questions, I think for me at least, it makes it be easier to write the discussion as well, because I've already thought about what the implications would be when I designed the questions, rather than sort of like letting it take me to where it wanted to.
That's great.
At what point do you actually do the pre-registration process? Like, do you do pilot studies first, or do you do some field work first? Like, because I know when you get out of the field, things can often really, really change. So yeah, at which point do you do it?
Yeah, I mean, it depends on how confident you are and how, like if you're doing something in a system that you've worked in before and you're reasonably aware of, I think doing it up front wouldn't be ridiculous. I think in most cases, a pilot study would be very wise, before you pre-register, and then you just include reference to that when you pre-register it, because data's never exactly what you want it to be.
Thanks, Hannah. Really nice talk. Very interesting. Just curious on following up from that question. Actually, as you know, deal with more the data longer, and you learn more either through fieldwork or working with the data, or just a PhD students who just started on these things, and then you learn more things through the day. The things changes across a long time, and what's the best practice now for if let's say you're just learning more and more over the days and new methods coming up, showing up on how to analyze the data and whether Bayesian statistics methods package shows up, and at what point is a good point to like registration and then keep that pre-registration and keep that, or at what point is deviation from your original decided methods still a good practice, or not trying to fish out data from that, or how do we weigh this?
I think it's really tricky in the context of particularly students, but realistically, we're all learning new approaches as we go. I mean, we should be, ideally. I think that my main recommendation would be to just do reasonably small scale pre-registrations. Like, don't pre-register your whole PhD up front. You'd be shooting yourself in the foot. But like, once you've got a good idea about what you're doing in a particular study, you pre-register that. And I would say, this is my rule of thumb that nobody agrees with, so take it with a very large grain of salt. Don't pre-register more than 2 analyses. And then you can do as many other analyses as you want, and you just say, these are the ones I pre-registered, and that's what the results are, and you have to present them in full. And then you include all of the other analyses you wanted to do as well. And they just have to take them with a little bit more grace.
And we've changed analysis from pre-registered and never had I haven't done it a million times, but you just go, we plan to do this based on this assumption.
Yeah, that's
That was a
wrong.
load of
Yeah.
that assumption, so now we've changed our approach to this. So transparency is, like, it's not meant to restrict you, it's meant to promote transparency.
Yeah, exactly. It doesn't feel nice to have to write, I thought that this made sense and I was wrong because this random effect structure just is nonsense. But that's been the true thing for us a couple of times. And I think it's actually good to have it out there in literature.
Has anyone ever had pushback for changing?
I haven't seen it, but
No, there's not.
possibly.
But if some of us have changed and never had pushback, then that's a good sign that you're not sort of trapped in this, oh no, me from a year ago model of.
It is 12 o'clock, so Hannah, how long are you here? How can people get in contact with you?
I'll be here all day somewhere. You can send me an email or just find me around. Yes, I'll be here all day today. I'll be here directly after this. I'll probably go out to lunch somewhere. I love company. So if anybody wants to ask more questions or talk about research in general, I'll be around. But yeah, always, if you email me at any point, I'll hopefully respond quickly, but definitely I will respond.
Please join me in thanking Hannah.